Hi, my name is Aisya, and for this video I'm going to explain subtopic 9.3, which is fertilization and fetal development. First, we are going to take a look at fertilization. Fertilization is a process of the union of haploid male and haploid female gametes. The importance of fertilization is to combine haploid set of chromosomes from male and female gametes to form diploid cell, which is 2N. And the second one is to activate egg through the contact of sperm head with egg membrane, thus triggering the embryonic development. There are four stages of fertilization. The first one is capacitation, the second one is acrosomal reaction, the third one is the fusion of sperm head membrane and secondary oocyte, and the last one is cortical reaction. The first step in fertilization is capacitation. During capacitation, sperm will undergo physiological changes where the coating material of the sperm will be removed. This process is stimulated by secretion of female reproductive tract. This process will establish sperms for acrosomal reaction. It also increases the sperm motility and function. The second stage is acrosomal reaction. Acrosomal reaction happens when sperm contact with zona pellucida. Then it triggers the exocytosis of acrosome to release hydrolytic enzyme. Sperm will be able to penetrate to zona pellucida as the hydrolytic enzyme digests the zona pellucida. The third stage of fertilization is fusion of sperm head membrane and secondary oocyte. As the tip of acrosome contact with the oocyte membrane, it will lead to the fusion of sperm head membrane and oocyte. Sperm nucleus will enter the cytoplasm of ovum, then will trigger the depolarization of secondary oocyte membrane. Then it will lead to cortical reaction. Secondary oocyte will complete meiosis II to produce ovum and polar body. The last stage of fertilization is cortical reaction. During cortical reaction, cortical granule of the egg will release enzyme that will harden the zona pellucida layer. This is to prevent additional sperm cell from fusing with the egg's plasma membrane. Later, envelope of nuclei from sperm and ovum will be dispersed. Male and female pronuclei will fuse to form diploid zygote. Zygote that form will be ready for the first cell division. After fertilization, a woman goes through pregnancy or gestation where she carries the developing individual in her uterus for 38 weeks before giving birth. Human gestation is divided into three periods of 3 months called trimester. 1 until 3 is the first trimester, 4 till 6 is the second trimester, 7 till 10 is the third trimester. Next is human embryogenesis. Human embryogenesis is a process by which embryo is formed and developed. Zygote will undergo cleavage and cell differentiation. Cleavage is a rapid mitotic division. First division of zygote is 24 hours after fertilization and it completes about 30 hours after fertilization. During the second division of zygote, the second cleavage is complete and there are 4 cells. During the third division of zygote, it produces 8 cells with 2-tier level of 4 cells. As the cleavage continues, the zygote develops into a bulk of cells passing down the oviduct to the uterus. The morula travel to the uterus around 3 to 4 days after fertilization, and at about 4 days after fertilization, a fluid-filled space called blastocoel cavity appears and the morula becomes blastocyst. During implantation, trophoblast will secrete enzyme to digest endometrium. As the endometrium has been digested, inner cell mass will be penetrated, resulting trophoblast to project into endometrium. Blastocyst then embedded into endometrium. Cells in the inner mass differentiate and form two layers of embryonic disc, which is epiblast and hydroblast. Finger-like projection of trophoblast lead to the erosion of capillary and blood will split out and surround the trophoblast. This will provide nutrients for the development of blastocysts. Gastrulation is a formation of gastrula from blastocysts involving the rearrangement of the cell of blastula that will produce three germ layers. Epiblast will give rise to ectoderm, mesoderm, and endoderm. All of these three layers will be surrounded by extra embryonic layer. Gastrulation involves the migration of cells where epiblast will be pushed inward, blastocore will be disappear, and archenteron will form. After gastrulation, there will be organogenesis. Organogenesis is a series that involves in development of organ and organ system from three germ layers, which is ectoderm, mesoderm, and endoderm. Process involved in forming neural tube and notochord is neurulation. Neurulation is a process to form nerula from gastrula. This process involves formation of neural tube from neural plate.
Neurulation is the formation of a thickened area of cells, called the neural plate. The neural plate forms at the cranial end of the embryo and grows in a cranial to caudal direction. The cranial or head end of the neural plate indicates the region of the future brain, and the narrower caudal or tail end represents the future region of the spinal cord. By the end of the third week of development, the lateral edges of the neural plate become elevated and move together to form the neural folds. The resulting space, created by the folding of the neural plate, is called the neural groove. The neural folds fuse together, and the neural plate transforms into the neural tube, the precursor to the central nervous system. Fusion of the neural tube usually begins in the middle of the embryo, extending in both cranial and caudal directions. During the closure of the neural tube, cells on the crest of the neural folds detach, forming a new cell population called the neural crest. These cells contribute to the formation of the peripheral nervous system. Once the neural tube has completely fused, the process of neurulation is complete.
For extra-embryonic membrane, there are four membranes that support the developing of embryo. The first one is chorion, the second one is amnion, the third one is yolk sac, and the last one is allantois. There are four extra-embryonic membrane. The first one is chorion. Chorion is developed from trophoblast. It is completely surround embryo and other extra-embryonic membrane. The outer layer will form chorionic villi where oxygen gas and nutrients are supplied to the fetus. It also forms fetal portion of placenta. The second one is amnion. Amnion formed from epiblast. It encloses embryo in fluid-filled amniotic cavity. It protects embryo from drying out and cushions it against mechanical shock. The third one is yolk sac. It formed from hypoblast. It encloses another fluid-filled cavity with no yolk. In human, it is small. It is site for early formation of blood cells. The last one is allantois. It is outpocketing from embryo's gut. It will incorporate it together with yolk sac into umbilical cord. It also will form blood vessels that transport respiratory gases, nutrients, and waste between fetus and placenta. Next, we will overview placenta. After 10 weeks of pregnancy, placenta will take over from corpus luteum to secrete progesterone, estrogen, and human chorionic gonadotrophin (HCG). Function of placenta. In terms of nutrient and digestion, placenta will transport nutrients such as glucose, amino acid, fatty acid, electrolyte, vitamin, and water through active and passive transport. Placenta also acts as storage of nutrients during early pregnancy. It is important to accommodate increase of fetal demand later in pregnancy. For endocrine function, placenta will secrete hormone to support development of fetus, for example, progesterone and estrogen. This hormone will prevent ovulation, prevent menstruation, develop mammalian gland, maintain and develop endometrium lining. It also will mediate transmission of maternal hormone into fetal blood and vice versa. For immunity, placenta will enable transfer of antibody, called as natural passive immunity. It also will act as a physical protective barrier. It will prevent pathogen from enter the fetal blood, prevent mixing of maternal and fetal blood, prevent some toxic to enter the fetal blood, and lastly block large and highly ionized molecule cross. For respiration, it will enable gases exchange between maternal blood and fetal blood. And lastly, for excretion, it will enable removal of waste products such as urea, ammonia, and uric acid. However, placenta does not completely stop all of the substance to across it. Certain viruses such as HIV, rubella virus, and Rh antibody potential can damage and cause death to embryo. Abnormalities that can occur. The first one is a child with anencephaly born without scalp for of cranium, brain hemisphere, and cerebellum. This condition occurs due to the abnormalities during gestation. The life expectancy for those who survive is only a few hours or days. The second one is thalidomide baby. This is caused by thalidomide drugs taken. It causes malformation or absence of thumbs, ears, as well as dislocation of the hip, heart defect, and malformed intestine. Fetal alcohol syndrome. Fetal alcohol syndrome is a condition in a child which is result from the alcohol exposure during mother's pregnancy. Fetal alcohol syndrome causes brain damage and growth problem. A child with fetal alcohol syndrome will have small eyes, thin upper lips, a short upturned nose, and a smooth skin surface between nose and upper lips. They will also have deformities of joints, limbs, and fingers. They will experience slow physical growth before and after birth. They also have vision difficulties and hearing problem. Lastly, they will have intellectual disability, learning disorder, and delayed development. Lastly, we will go through fetal growth and development. During the first trimester, embryo will undergo embryogenesis, implantation, gastrulation, organogenesis, and neurulation. The major structure that present is heartbeat, limb will be appear, brain development, muscle differentiation, gonad distinguished to ovary or testis, sex can be determined at 3 months, notochord degeneration, and limb gland generate. There also will be development of embryonic membrane, placenta will grow, and trophoblast will secrete HCG to maintain structure of corpus luteum. During the second trimester, fetus will continue to grow in length rapidly, and then fetus will actively move. Organogenesis will continue, and there will be presence of human appearance such as eye, nose, and ear. Hormonal level will stabilize as HCG decline. Corpus luteum will be degenerate as the placenta is fully formed. Placenta will secrete progesterone to maintain the pregnancy. During the third trimester, fetus will grow to about 3 to 4 kg in weight and 15 cm in length. Fetal activity will decrease due to limited space. Maternal abdominal organ will become compressed and displaced. Head position will be downward before labor. Fetus will be terminated during birth.